move forward, proceed with water soluble vitamins. So today we'll basically be focusing on water soluble vitamins, but after I finish with vitamin K. Okay, so introducing you to the core features of Oracle. Here at Oracle, we have live classes to elevate your learning experience. Then we have recorded classes where you have the facility to access any of the subjects as per your timings according to your own convenience. And apart from that, we also provide you with notes based on the latest CBME syllabus, and we also give you MCQs where you can assess your knowledge. And apart from that, a unique feature of Oracle is that we have personalized counseling sessions. So anytime if you feel like you need some extra support and guidance from our end, so you can book a slot with us for that extra support. So apart from the academic support, we are also here to give you non-academic support as well. Then we have three different subscription plans at Oracle. That is basic, standard, and premium, which comes for 3, 6, and 12 months consecutively. Then basic is for 15,000, standard is for 30,000, and you have the premium plan, which is for 50,000. But apart from that, you can get additional 20% off in any of the subscription plans if you use my coupon code as displayed here on the screen. Then in the premium plan, you also have the facility of live doubt clarification, as well as there is one-to-one personalized guidance. Also, we provide you with notes in the premium plan as well. Then introducing you to the internal exam marathon series. So this series has been specifically curated to target your university exams, which are approaching soon this time. So we are targeting all the important topics here in this series in a rapid revision kind of a way, where we focus on touching at least the periphery of all the topics. Then we'll also be covering volatile facts. Apart from that, multiple choice questions will also be dealt so that we can assure you top-notch grades in your exam. So before I begin with water-soluble vitamins, I'll take up vitamin K, which is a fat-soluble vitamin in our last lecture. Because of time, this was left. So let me take vitamin K first. So it is a naphthoquinone derivative with a long isoprenoid side chain. The structures are not very important, but sometimes you might get an MCQ. And apart from that, also, if you have the structure in mind, like right now, I'm giving you the structure, you need not like learn the structure because it's never really asked because you're medical students, not chemistry students. But if you have, while I'm giving this lecture and somewhere at the back of the mind, you have this idea now, when you get these things in questions or in reactions, so you know how it is taking place. You have a slight rough idea of the mechanism. Therefore, the structure becomes important, okay, for the sake of understanding. So it is a naphthoquinone derivative with a long isoprenoid chain. Okay, now depending upon the length of the side chain, you have three different forms, that is pheloquinone, menaquinone, and menadione. So in pheloquinone, you have 20 carbon side chain, which is vitamin K1. It is derived from plant sources. Menaquinone, it has 30 carbon side chain, which is known as vitamin K2, and it is synthesized by intestinal flora. And then you have menadione, which is a synthetic compound. Okay, it is similar in structure to vitamin K, and it is water soluble. So here you can see naphthoquinone derivative, and this is the side chain. So depending upon the length of side chain, we have pheloquinone, where there is 20 carbon length. Then you have menaquinone, which is 30 carbon, and menadione, you have just one hydrogen. Okay. Now the functions are important. So we said that your fat soluble vitamin, if you remember in the introductory part, I told you that they are involved in various roles, like your vitamin A, it had a role to play in vision, in reproduction, in maintenance of the epithelium, in growth and development. Then your vitamin D, it was involved in bone mineralization and tooth development and all that. And your vitamin E, it was an antioxidant. But this is the only fat-soluble vitamin, that is vitamin K, which is a coenzyme role, okay? Now, it helps in coagulation by gamma carboxylation. So, your vitamin K, it is involved in clotting process and coagulation. How? By gamma carboxylation. So first we need to know that what is gamma carboxylation. So gamma carboxylation, what is carboxylation? I hope you all know. It is the addition of the carbon. Okay, so in gamma carboxylation, there is carboxylation of glutamyl residues at gamma carbon in proteins to form gamma carboxyglutamate. Okay, so what is this? If you have a look at this diagram here, so in this polypeptide chain that is shown here in green, they have actually enlarged, zoomed in this glutamyl residue. Okay, here. This glutamyl residue has been enlarged. This you can see. Now it says that there is carboxylation of this glutamyl residue at the gamma carbon. This is your gamma carbon, okay? This is your alpha, beta, delta, gamma. So this is your gamma carbon, and at this gamma carbon, what is happening? Carboxylation is happening. So you see this carbon dioxide has been added here at the gamma carbon here. Okay, so this addition of a carbon dioxide at the gamma carbon of a glutamyl residue in a polypeptide chain, this is known as gamma carboxylation, and this is brought about by vitamin K. Okay, so you need vitamin K for this reaction. Okay, so the enzyme involved is gamma glutamyl carboxylase. So in a protein to the glutamic acid, as you can see here, this glutamyl residue, what has happened to the gamma carbon, a carbon dioxide molecule has been added. Okay, so this is known as gamma carboxylation. And this glutamyl residue now becomes a gamma carboxyl glutamyl residue, also known as GLA residue. Okay, so that was briefly about gamma carboxylation. This diagram in itself is very explanatory. I'll repeat once again. You can see here this polypeptide chain, and this polypeptide chain to the glutamyl residue. At the gamma carbon of the glutamyl residue, a carbon dioxide molecule has been added. This is brought about by the enzyme gamma glutamyl carboxylase. And for this reaction, what is needed? Your coenzyme is vitamin K. So vitamin K has the role of a coenzyme here. And what do you end up with? You end up with GLA residues. So that was gamma carboxylation for you. Now gamma carboxylation requires gamma glutamyl carboxylase, oxygen, carbon dioxide, and hydroquinone form of vitamin K, which gets oxidized to epoxide form. Okay, so for this reaction, you need specifically the hydroquinone form of vitamin K. And before the reaction, what is happening? It is in the hydroquinone form, and after when this reaction has been completed, it is oxidized to its epoxide form. Now, before causing gamma carboxylation, vitamin K is in hydroquinone form. After gamma carboxylation, it is converted into epoxide form. So also what the text says now, vitamin K epoxide reductase is the enzyme which converts the epoxide form of vitamin K to the hydroquinone form. Okay, for the reaction to take place, for gamma carboxylation to take place, you need hydroquinone form. But after the reaction, it has been converted into epoxide form. Your hydroquinone form has been converted into epoxide form. Now, this epoxide form has to change back into hydroquinone form. For this reaction, you need VKOR. Okay, a VKOR, that is vitamin K epoxide reductase, can be inhibited by drugs like warfarin and ducomurol. Okay, so they are the inhibitors of vitamin K epoxide reductase. So if vitamin K epoxide reductase is inhibited, you will not have hydroquinone form of vitamin K, and ultimately what will not happen? Gamma carboxylation will not take place. Okay. Now, which are the proteins that are gamma carboxylated? So they are your factor 2, which is the prothrombin, factor 7, factor 9, and factor 10. The GLA proteins are the binding sites for calcium binding. So calcium binding is a very important part of coagulation. You must have studied about this in physiology. So we're not going into coagulation. Do you know that GLA proteins, that is gamma carboxylated proteins, they are the binding sites for calcium. See, as you can see here in this diagram, you have your glutamic acid residue. Now, after it undergoes gamma carboxylation, you can see here, this is a GLA protein. This GLA protein, this gives binding site for calcium, as you can see here in the diagram also. So once the factors are gamma carboxylated, then only they can bind to calcium. Okay. So gamma carboxylation becomes very important for calcium binding, and calcium binding is very important in the clotting process. Now, other proteins that are gamma carboxylated, they are protein C, protein S, osteocalcin, that is synthesized by osteoblasts and seen in bones, and then you have nephrocalcin, which is a protein seen in kidneys. Moving on to the deficiency of vitamin K, so there's increased bleeding time. So obviously what will happen if you do not have carboxylated factors, what will happen? Calcium binding will not take place, so coagulation process will be affected. And ultimately what is happening? There is increased bleeding time. There is increased growth hormone time. Vitamin K deficiency is important in premature or neonates because this immaturity of liver, the sterile gut, okay, the gut is still not very developed. So vitamin K is synthesized, as we all know, by the intestinal flora. So that synthesis will not take place. Breast milk is poor in vitamin K and there is poor placental transport. But talking about the toxicity of vitamin K, hemolysis, jaundice, hyperbilirubinemia, kernicterus, and brain damage. Source your green leafy vegetables. They are a good source of vitamin K. Even if the diet does not contain the vitamin, intestinal flora will meet the daily requirement as long as absorption is normal. And RDA, that is 50 to 100 mg per day, is required. It is usually available in the normal diet. And if not, it will be compensated by the intestinal flora. So with this, we come to the end of vitamin K. Moving on to vitamin B1. Okay. So before I begin with our next topic, that is vitamin B1 thiamine, I'd like to explain you that where actually we are right now in vitamins. So I told you that vitamins are divided into water soluble and fat soluble. I hope the diagram is clear. Okay. Then we have taken the fat soluble vitamins. That is, we've taken vitamin A, vitamin D, vitamin K, and vitamin E. So we're done with this part. Now moving on to the water soluble vitamin, which is our topic for today. So in water soluble vitamin, you have the B complex and you have the non-B complex. So non-B complex, you only have vitamin C in this, which we'll be taking in our next class. But today we'll be taking vitamin B complex, okay? And in B complex, again, you have energy releasing B complex vitamins, you have hematopoietic, and then you have others. So beginning with energy releasing vitamins of the B complex. So that is, today we'll be doing B1, that is thiamine, followed by B2 riboflavin, the niacin, biotin, and pantothenic acid. And in hematopoietic, we have folic acid and cobalamin. And in others, we have pyridoxine, and pyridoxamine, which we'll be taking in the next lecture. So for today, let's begin with vitamin B1, that is thiamine. Okay, so beginning with thiamine, so its structure, it contains a pyrimidine ring in its structure. The source is unpolished rice, wheat, parboiled rice, and yeast. Thiamine is present in the aleurone layer. So your rice has an outer layer, okay? And in between this outer layer, in between is present a very thin layer. A thin layer is present in between, which is known as aleurone layer. And this is rich in thiamine, okay? But this layer, this is lost in polished rice or wheat. And what is par-boiled rice? That is partially boiled rice while it is still in the husk. Okay, so it is an old technique of preserving the aileron layer, as this aileron layer is rich in thymine. Okay, so these were the sources of thymine. Now moving on to its function. So talking about its coenzyme role, the active form of thymine is thymine pyrophosphate, TTP, or thymine diphosphate. Now the enzymes that require TTP. So your pyruvate dehydrogenase, so alpha keto dehydrogenase, branch chain keto acid dehydrogenase, and ketolase. They require thymine. So your pyruvate dehydrogenase, this is involved in the link reaction, alpha ketoglutarate dehydrogenase, which is involved in TCA. And you see, both these enzymes, they are involved in energy production, okay, in ATP production. They are involved in ATP production. So if there will be TTP deficiency, what will happen? The activity of these enzymes will be low, and as a result of which there will be decreased ATP production. Okay, so these are the energy requiring enzymes. Therefore, they're classified into energy requiring vitamins. Your TTP. Then branch chain keto acid dehydrogenase. We've already seen this in when we were studying about branch chain amino acids. Okay, it was the deficiency or defect of this enzyme. It was related with maple syrup urine disorder. Then trans ketolase, we've studied about this in non-oxidative phase of HMP. We've actually spoken about all these enzymes before as well. Now moving on to the deficiency of vitamin B1. This is very important. So berry is very important. You often get asked about this as short notes. You might be asked about a specific kind of berry, wet berry, dry berry, infantile berry, or you might be asked about the entire berries. You might get a question like, what is berry? Define its type, or about a single type as well. So this is very important. Berry is very important. So adult berry, you have adult beriberi and you have infantile beriberi. So speaking about adult beriberi, it is again classified into wet and dry. So your wet beriberi, it is associated with cardiovascular manifestations. So there's cardiac failure, dyspnea, there's cardiomegaly, there's peripheral edema, and there's pulmonary edema. Moving on to dry beriberi. So this affects the peripheral and central nervous system. So as a result of which there is loss of reflexes, especially in the lower limb. There are also muscle cramps, and in extreme cases, there might also be muscle atrophy. Now, moving on to infantile beriberi, it is seen in nursing infants whose mothers are deficient in thiamine. And now there's another kind of beriberi, which is cerebral beriberi. It is also known as Wernicke's Korsakoff syndrome. Okay, now what happens is there is something known as Wernicke's-Korsakoff encephalopathy. Okay, so in encephalopathy, you have the manifestations that is, it is manifested in the CNS. It is present with horizontal nystagmus. That is, there is rapid, repetitive movement of the eye. Then there's ophthalmoplegia, there's truncal ataxia, there's confusion, memory loss, and psychosis. So this confusion and memory loss and psychosis, this is present in Wernicke's-Korsakoff syndrome. And these are the features of Wernicke's-Korsakoff encephalopathy, okay? So what is happening in Wernicke's-Korsakoff syndrome? That is, you have all the features of Wernicke's-Korsakoff encephalopathy, plus there is confusion and memory loss and psychosis. Okay. And if we talk about ophthalmoplegia, what is this? This is drooping of the eyelids. And your nystagmus, this is rapid, repetitive, involuntary movement of the eye. So what are the different types of berries that we have? You have adult, which is divided into dry and wet. Then you have infantile, and then you have cerebral beriberi. Talking about the thiamine toxicity, as such, no toxicity has been reported. In RDA, it is 1 to 1.5 mg per day requirement. Moving on to our next vitamin, that is riboflavin. Okay. So beginning with the introduction, this is a light sensitive vitamin, therefore supplemented during phototherapy. Because during phototherapy, it is lost. This is destroyed from our body. Therefore, you need to supplement this during phototherapy as it is a light sensitive vitamin. A pigmented vitamin, also known as Warburg yellow enzyme, and it gives urine a yellow color. This is also the reason why on taking B complex vitamins, the urine becomes yellow. Okay. Therefore, on taking B complex vitamin supplement, your urine turns yellow. And it is a heat stable vitamin, so therefore cooking food does not destroy this vitamin. Now moving on to its coenzyme role. The active form of riboflavin, it is FMN or FAT. So your mononucleotide is FMN and FAD is flavin adenine dinucleotide. So if we talk about the structure of riboflavin, again not required, but so that you understand what is FMN and FAD, I'll just tell you the structure of riboflavin. It has an isoalloxanthine and a ribitol sugar. So, okay, so you don't have to like remember these names, so just so that you understand the structure. I'm just showing you this, okay? So this is your isoalloxane and this is your ribitol, okay? So this forms what? Which is known as riboflavin, okay? This forms riboflavin here. This together, this will form riboflavin. Now, to this riboflavin, what happens when you add a phosphate? When a phosphate is added to this riboflavin, it becomes what? Riboflavin plus a phosphate, it becomes FMN. Okay, so just for the sake of understanding, I'm telling you this. So what is FMN? It is the active form of riboflavin. So you can see here in this diagram, this is your riboflavin. And to this riboflavin, has been added? A phosphate has been added. So this becomes riboflavin plus phosphate. They together form FMN. Okay. And what is FAD? Now, FAD is that you add an adenosine monophosphate, that is AMP, to an FMN. Okay. So, I'll explain you this here. See, this was your FMN. Now, to this FMN, a phosphate, a ribose sugar, and an adenine. What is this? You have a pentose sugar, you have a nitrogenous base, and you have one phosphate. So what is this? This is an adenosine monophosphate. This is an AMP. Okay, we all know this, that pentose sugar nitrogenous base and a phosphate, single phosphate. This is adenosine monophosphate. Okay, no big deal. Now this adenosine monophosphate and this FMN combined together, when they join together, they form what? FAD. So what all do you have in FAD? In FAD, you have these structures as shown here. Okay, understood? This is exactly what this diagram also shows. See, I hope it's clear. So you can see this and this diagram as well. What do you have? You have here, you have this adenine and you have this pentose sugar and you have this phosphate. So this is AMP and this another phosphate and this riboflavin. This was FMN. Now FMN and AMP together, they form FAD. Okay, that is flavin adenine dinucleotide. Okay, so this is the active form of riboflavin, that is FMN and FAD. Now moving forward, FMN and FAD are capable of reversibly accepting two hydrogen atoms and form FMNH2 and FADH2. You all know this. Now, FMN and FAD exist in tissues tightly bound to enzymes that catalyze the oxidation and reduction of substrate. So that is known as flavoproteins. So what are your flavoproteins or flavoenzymes? They are enzymes that contain riboflavin. So flavoenzymes, they are enzymes that contain riboflavin. So what is happening is your FMN or your FAD, they are found attached. They're tightly bound to enzymes. Okay, the enzymes which are involved in oxidation and reduction of substrates like your NADH dehydrogenase or succinate dehydrogenase. Your NADH dehydrogenase, you studied about this. This is the complex 1 of electron transport chain FAD. This is complex 2 of electron transport chain. So NADH dehydrogenase, it is bound with FMN, or succinate dehydrogenase is bound to FAD. Okay, so your FAD, FMN, they are found bound to enzymes which are involved in oxidation or reduction of substrate. Moving on to the deficiency. Initially, it is asymptomatic and later on manifestations appear like chillosis, that is twitching at the corner of the mouth, glossitis, that is tongue appearing smooth and dark, and angular stomatitis, that is inflammation of the corners of the mouth. And toxicity has not been reported yet, talking about the RDA, which is 1.5 mg per day. So that was about riboflavin. Moving on to niacin, also known as vitamin B3. Okay. So niacin is an endogenously synthesized vitamin from amino acid tryptophan. We actually studied this while we were studying tryptophan metabolism. We had seen how during the metabolism of tryptophan, there was this intermediate known as anthranilic acid, and that was going into NAD pathway. It is not strictly a vitamin. Now, talking about its coenzyme role, its active form is NAD plus and NADP plus. NAD plus is nicotinamide adenine dinucleotide. NADP is nicotinamide adenine dinucleotide phosphate. So similarly, like I described you to the structure of riboflavin, this is also similar to it. Okay, and just that instead of riboflavin, you have niacin. Like your riboflavin was derived from ATP, this is also derived from ATP. Now moving forward to the enzymes that require NAD. So every dehydrogenase requires NAD with the exception of those enzymes that require FAD or NADP. So apart from the enzymes, dehydrogenases that require FAD and NADP, rest of all the hydrogenases, they require NAD. Now, NADP requiring enzymes. So they are requiring NADP. So they're requiring NADP and they will be generating NADPH. So you can either say NADP requiring or NADPH generating. They are glucose 6-phosphate dehydrogenase, 6-phosphogluconate dehydrogenase. If you remember, we've already studied these enzymes. These are the first two enzymes of your oxidative phase of HMP. Now, malic enzyme cytoplasmic isocitrate dehydrogenase. So these are the enzymes that require NADP. Now, NADPH requiring enzymes, almost all reductases require NADPH. That is your enoyl reductase, P2SIL reductase. Talking about them in fatty acid synthesis, okay? HMG-CoA reductase, this is required in cholesterol synthesis. Now, glutathione reductase, it is involved in free radical scavenging. Ribonucleotide reductase, it is involved in the conversion of ribonucleotide to deoxyribonucleotide. And folate reductase, which is involved in 1-carbon metabolism. So we've spoken about the NADP requiring enzymes and NADPH requiring enzymes. Now moving on to the deficiency of niacin, this is very important. So pellagra is very important. So basically the clinicals are more important than studying the structures and all. But you should know the active forms and the coenzyme forms, their coenzyme role, because while studying the reactions, you'll notice that they form an important part. Okay, so moving on to the deficiency, initially present with vague symptoms like lethargy, loss of interest in the surrounding, etc. And gradually it progressed to pellagra, which is a disease involving the skin, gastrointestinal tract, and central nervous system. So the symptoms or the manifestations of pellagra, it manifests through 4Ds. These are very important. What are the 4Ds of pellagra? It is dermatitis, diarrhea, dementia, and if untreated, death. So what is dermatitis? You have lesions on the sun-exposed area. So you have skin lesions on the photosensitive dermatitis. You have skin lesions on sun-exposed area. Okay, so which is known as Castle's necklace. Present in the collar of the neck, okay, because this is the area exposed to the neck, so it forms a necklace like a structure which is known as Castle's necklace. When you have diarrhea, dementia, and left untreated, there might be death. The corn is low in niacin and tryptophan, so corn-based diets they can cause pellagra, and also in regions where the staple diet is jowar. Okay, so jowar it has high content of leucine. And your leucine, this inhibits, it inhibits what? Inhibits QPRTase, okay, which is the rate limiting enzyme for niacin synthesis. So in also regions where jowar is the staple diet, you'll have what? You'll have a higher content of leucine, which will inhibit the niacin synthesis. So also you'll have pellagra. Now, other conditions associated with pellagra. So you have vitamin B6 deficiency. It inhibits the enzyme kynureninase, which leads to decreased niacin synthesis. If you remember when we were talking about tryptophan metabolism, there was this intermediate kynureninase, which was acted upon by kynureninase, and ultimately what were you getting? You were getting another intermediate known as anthranilic acid. Okay, and this anthranilic acid, it was going into niacin synthesis. So for this kynureninase enzyme to function, PLP was needed. So if there's deficiency of B6 or PLP, what will happen? Kynureninase will not be formed. Kynureninase will not work, and what will not be formed? Your anthranilic acid intermediate will not be formed, and ultimately what is happening? Your niacin synthesis is decreased. Now, Hartneff's disease, in this what was happening? There was tryptophan absorption defect. Okay, so your tryptophan was not being absorbed, which causes tryptophan deficiency, and which in turn will ultimately cause niacin deficiency, because your niacin is being formed from tryptophan only. So there's this very nice mnemonic that I've formed for you. Hartneff's disease, okay? Hartneff disease. Okay, so in this Hartneff, you have AR. So AR stands for autosomal recessive. T for there is transporter defect of tryptophan. NU because neutral amino acid transport defect. And P for you have pellagra-like symptoms. So if you remember this mnemonic Hartnup disease for Hartnub's disease, so from the Hartnub, at least these are the facts that you can remember very easily. Moving on to pantothenic acid. Okay, so pantos means everywhere, suggesting it is widely distributed in nature. So your pantothenic acid is widely distributed in nature, and endogenously it is synthesized in intestinal flora, by the intestinal flora. Now, just talking about the structure, again, the structure is not as such very important, but if I tell you about this structure, if I put this diagram here right now during the lecture, somewhere at the back of your mind, it will stick with you and you will be able to understand the mechanism of reactions. So it contains beta-alanine and D-pantoic acid in amide linkage. So what is your pantothenic acid? You can see this beta-alanine present here and pantoic acid here, and both of them, they are joined together by an amide linkage forming pantothenic acid. So this is your pantothenic acid. Now moving on to the coenzyme role of pantothenic acid. Your pantothenic acid, which is present in coenzyme A, very important, an acyl carrier protein. So your coenzyme A, it is involved in transfer of acyl group. Transfer of acyl group. And your acyl carrier protein, it is a domain of fatty acid synthase. That is fast. If you remember when we were talking about lipids and fatty acid synthesis, we had also spoken about FAS. Okay, so one of the domains of FAS, it has acyl carrier protein, and that acyl carrier protein contains pantothenic acid. And also coenzyme A contains pantothenic acid. Coenzyme A, it is a complex molecule. Here I put a diagram of coenzyme A. You can see there is beta-alanine And there's pantoic acid. So beta-alanine and pantoic acid together, they form pantothenic acid. Apart from pantothenic acid, your coenzyme A has beta-mercaptoethanolamine. It also has an ADP. So your vitamin B5 is present in coenzyme A and in acyl carrier protein. It is required for succinyl-CoA, acetyl-CoA, and HMG-CoA. So, succinyl-CoA and acetyl-CoA, they are present in TCA cycle. Your HMG-CoA, it is required for cholesterol synthesis, ketone body synthesis, and in glucine catabolism. Now, talking about the deficiency. So you have one disease, that is Gopalan's burning foot syndrome, manifested as paresthesia, that is burning lightning pain in lower extremities. Then there is staggering gait. These deficiencies manifestations are rare in human beings. The syndrome is seen during famine, in prison camps, in chronic alcoholics, and in some renal dialysis patients. So as such, this is very rare. Or in case of famine, in chronic alcoholics or renal dialysis, it can be manifested. That is your Gopalan's burning foot syndrome. So that was about B5, pantothenic acid. Now moving on to B7, that is your biotin. So this is our last under energy releasing B complex vitamins, that is biotin. So beginning with the introduction, evidin, which is a glycoprotein present in raw egg, it binds to biotin and prevents its absorption. So if you are consuming raw egg, you will have what? You will have evidin, and which will bind to biotin, preventing its absorption. So in that case, you can have biotin deficiency. So evidin can bind to biotin, leading to biotin deficiency. Otherwise, biotin deficiency does not occur naturally because this vitamin is widely distributed in food. So otherwise, biotin deficiency is rare and does not occur naturally unless and until you consuming raw egg. So it is very important that you don't consume raw egg. Moving on to the structure of biotin, it consists of an imidazole ring fused with a thiophene ring. You can see here, this is an imidazole ring which is fused with a thiophene ring with a valeric acid side chain. So in the side chain, you can see valeric acid here. Now the carboxyl of this valeric acid, it forms an amide linkage with the upsilon nitrogen of lysine residue in biotin dependent enzyme. So those enzymes which are biotin dependent, how are they engaged with biotin? So the enzyme which is biotin dependent, that is your ABC enzymes, the lysine residue in those enzymes. So the epsilon nitrogen of the lysine residue in the ABC enzymes, it forms an amide linkage with the valeric acid side chain of your biotin. Now talking about the active form of biotin, carboxy biocystin is the active form and biotin is the vitamin for carboxylation. So your biotin is involved in carboxylation reactions and so many examples of this we've already studied. That is your ABC enzymes that require biotin. And what are ABC enzymes? If you remember, I told you when we gluconeogenesis. So A stands for ATP, they require ATP. B stands for they require biotin, and C stands for they require carbon dioxide. Okay, and they are involved in carboxylation. So your pyruvate carboxylase, acetyl CoA carboxylase, propanyl CoA carboxylase, and methyl chronotonol CoA carboxylase. So these are all ABC enzymes. That is, they are involved in carboxylation and they require biotin. There are also certain biotin independent carboxylations as well. Okay. Now, other uses of biotin. The affinity of evidin to biotin is greater than most of the usual antigen-antibody reactions. Therefore, evidin biotin system is commonly utilized in detection of pathogens by ELISA. So this is another use of biotin. So apart from being involved in carboxylation reactions, it is also used in ELISA. So instead of radioactive labeling of nucleotides, biotin labeling of DNA is becoming popular. So your DNA is instead biotin labeled. Now talking about the deficiency, there are mental changes. So depression, hallucination can be its manifestations, scaling, seborrheic and erythematous rashes around nose, eyes, and mouth. So nothing much about biotin. So with this, we come to the end of energy releasing vitamins. So here, so we finished this part. We finished this part of vitamins. So for our next class, we left with hematopoietic and others in B complex, and then you have ascorbic acid, that is vitamin C in non-B complex. And with that, we'll come to the end of water-soluble vitamins. So if you have any queries, you can send them in the comments. With this, we come to the end of today's lecture. Okay, thanks for watching. See you in the next class.